In this module, we will delve into the structure and function of living cells within the human body. Once completed with this module, you should be able to discuss cell theory, describe cellular anatomy and physiology, explain cellular respiration, describe the cellular environment, describe cellular transport mechanisms, and discuss the process of cellular division. Remember that cells are the building blocks for living organisms. As discussed in an earlier module, when a number of specific cells are grouped closely together, they form tissues. Tissues that function together form organs. Organs form body systems, and these systems together comprise the human body. The individual cells within the human body can be very different and perform a multitude of functions that include movement, conductivity, metabolic absorption, secretion, excretion, respiration, and reproduction. Regardless of the cell's shape, form, and function, the basic structure and components of the body's cells are usually the same. To begin, every cell has a cell membrane. Think of the membrane as the skin or wrapper for the cell. The cell membrane defines the borders of the cell and encompasses the environment within the cell. The contents within this membrane and the cell are referred to as intracellular. Those substances outside this membrane are called extracellular. In the previous module, we spoke about phospholipids, which are a lipid that contains a phosphate group in its molecule. These molecules line up next to each other with the hydrophilic, water-loving phosphate group head on one side and the hydrophobic, fatty acid tails on the other. A second inverted layer, if you will, then combines with that first layer to create what is known as a lipid bilayer. The benefit of this structure is that phospholipids are selectively or semi-permeable that allows certain molecules to pass through to either enter or exit the cell. Those molecules that dissolve in lipids, fat, pass through easily, such as oxygen, carbon dioxide, alcohol, and urea. Other molecules that are either larger or dissolve in water, such as glucose, sugars, amino acids, nucleic acids, proteins, and certain ions, such as sodium or potassium, cannot pass through this membrane easily. The cell membrane also contains proteins, known as membrane proteins, that serve a few different functions. Channel proteins create a channel or tunnel for certain molecules to enter and leave the cell. Water, for example, cannot pass through the phospholipid bilayer because the inner structure is hydrophobic. It repels water. Channel proteins are necessary for water to move into and out of the cell. There are also channel proteins that form what are known as gated ion channels. These specialized proteins open and close to allow charged ions from dissolved salts, such as sodium, potassium, calcium, and chloride, to pass through the cell membrane. A ligand-gated ion channel opens and closes in response to the binding or unbinding of a small molecule to the channel protein. A voltage-gated ion channel reacts to voltage across the membrane that is sensed by charged segments of the channel protein. As you will see, these channel proteins are essential for proper cell functioning, and many of the interventions paramedics can perform in the field work by impacting the functioning of these channel proteins. If you have ever heard of a calcium or a sodium channel blocker, that would be an example of a medication that works by impacting this cell membrane feature. In a previous module, we talked about enzymes and how they are catalysts for organic chemical reactions. Cell membranes can have protein enzyme receptors within the cell that allow enzymes to bind to the cell membrane and function as a catalyst for a specific chemical reaction within the cell. Some proteins on the cellular membrane act as receptor sites that allow certain various molecules to attach to the exterior of the cell. When this binding occurs, it then initiates a change in cellular function. Narcotics and narcotic antagonists work by binding to narcotic receptor sites on cells. So that the body knows which cells are which, cells can have identifier proteins that function to help the immune system determine which cells are part of the organism and which are intruders that should be attacked and eliminated. Somewhat similar to channel proteins are carrier proteins. They are similar to channel proteins in that their function is to allow molecules to move across the cell membrane. Rather than this being a passive process, however, carrier proteins actively move molecules across the cell membrane into or out of the cell. A good example of a carrier protein is the sodium-potassium pump, which moves potassium into the cell while moving sodium out of the cell. There are also membrane proteins that help determine the shape of the cell and others that facilitate the adherence of membrane proteins between adjacent cells to form tissue. In just a little bit, we will talk about organelles within a cell. These very important structures exist within the cell by floating within a fluid-like material known as cytoplasm. In addition to providing an intercellular environment for these various structures within a cell, the cytoplasm provides physical protection, like a shock absorber, if you will, from damage for these structures and the cell's genetic material given movement or collisions with other cells. The cytoplasm also serves as a medium for the transport of both genetic material and the products of cellular respiration. The composition of cytoplasm includes a substance known as cytosol, which is predominantly water with some protein strands that help keep the organelles in place, given that the organelles are not neutrally buoyant given changing concentrations of solutes and the impact of gravity. The cytoplasm also has what are known as cytoplasmic inclusions, which are non-soluble molecules that float within the cytoplasm, such as glucose, amino acids, fatty acids, ions, lipids, proteins, adenosine triphosphate, and waste products of cellular metabolism. Some of these molecules, like glucose and fats, are actually stored within the cytoplasm itself so that the cell has the fuel it needs for cellular respiration. The nucleus within a cell is typically the most prominent organelle within the cell. While not all cells have a nucleus, those within animals, including humans, do. Just in case you were curious, cells with a nucleus are called eukaryotic cells. Cells without a nucleus, such as bacteria, are called prokaryotic cells. If you want to think of a cell as a miniature living organism, the nucleus would be the brain of the cell, as it is the control center for the cell. The nucleus controls the metabolic activities of the cell while also storing genetic information, producing necessary enzymes for cellular activities, assisting in cell reproduction, and regulating the synthesis of proteins within the cell's cytoplasm. The structure of the nucleus is actually somewhat similar to the cell itself in that it has what is known as a nuclear envelope built from phospholipids that form a lipid bilayer that surrounds the interior contents of the nucleus. This nuclear envelope not only provides the shape of the nucleus, but it also contains nuclear pores that assist in regulating the flow of molecules into and out of the nucleus. The nuclear envelope is also connected to another cellular structure known as the endoplasmic reticulum, which will be discussed in just a little bit. The nucleus contains chromosomes, which are protein-based thread-like structures that contain the blueprint for that particular organism with strands of deoxyribonucleic acid, DNA. When a cell is not dividing, not reproducing itself, those chromosomes exist as long entangled structures called chromatin. They technically do not become chromosomes until the chromatin condenses down prior to cell division. Chromosomes are necessary to ensure that DNA is accurately copied and distributed during cellular reproduction. The nucleus is filled with a gelatinous, semi-aqueous material called the nucleoplasm, which is similar to the cell's cytoplasm. This nucleoplasm is made predominantly of water with enzymes, dissolved salts, and other organic molecules within it, and it supports the movement of materials such as enzymes, DNA, and ribonucleic acid, RNA, subunits, while also protecting the contents of the nucleus and helping it maintain its shape within the cell. The nucleus itself also contains a structure called the nucleolus, which is a dense structure without an exterior membrane composed of RNA and proteins. Another cellular organelle we will be discussing in just a bit is a ribosome. The nucleolus is critical for the creation of ribosomes, and it contains nuclear organizers, which are parts of chromosomes that contain the genes necessary for ribosome synthesis. The nucleolus also transcribes and assembles ribosomal RNA subunits, which join together within the nucleus during protein synthesis, which, as we previously mentioned, is yet another duty of the nucleus. Aside from the nucleus, there are other organelles within our cells. Because we just discussed the nucleus and the endoplasmic reticulum is attached to the nucleus, let's begin with that structure. The endoplasmic reticulum is a structure of tubules and flattened sacs that plays a key role in the production, processing, and transport of proteins and lipids. The endoplasmic reticulum is divided into two parts, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum is called that because it has ribosomes attached to it. Given these attached ribosomes, the rough endoplasmic reticulum is a place where proteins are synthesized and membranes are produced. In pancreatic cells, the endoplasmic reticulum produces insulin. The smooth endoplasmic reticulum is the area where carbohydrates and lipids such as cholesterol and phospholipids are synthesized. The breakdown of alcohol in some drugs occurs within the smooth endoplasmic reticulum of liver cells. The contraction of muscles is assisted by the smooth endoplasmic reticulum within muscle cells, and the smooth endoplasmic reticulum in brain cells synthesizes male and female hormones. The smooth endoplasmic reticulum also serves as a traditional area for transport vesicles, allowing proteins and membranes made by the rough endoplasmic reticulum to be transferred to other locations. We just mentioned that the rough endoplasmic reticulum has ribosomes attached to it. Ribosomes are also found within the cell cytoplasm, or more specifically, the cytosol, which is a fluid component of the cytoplasm. Ribosomes are essentially comprised of small ribosome subunits of RNA and protein produced in the nucleus that, when pieced together in the cell's cytoplasm, translate messenger RNA into a polypeptide chain during protein synthesis. Those ribosomes within the cytoplasm will typically produce proteins that are utilized within the cell itself, while bound ribosomes, those on the rough endoplasmic reticulum, usually generate proteins that are exported from the cell or are included in the cell's membranes. While these two functions may sound specialized, free ribosomes and bound ribosomes are interchangeable, and the cell can change their numbers given the metabolic needs of those cells. Golgi apparatus, also called Golgi complex or Golgi body, are membrane-bound organelles in eukaryotic cells that are made up of a series of flattened, stacked pouches in a semicircular bent formation called cisternae. One way to think of Golgi apparatus is that they are the manufacturing and shipping centers of the cell. Golgi apparatus commonly takes simple molecules synthesized and secreted by the endoplasmic reticulum, such as proteins and phospholipids, and combine them to make molecules that are more complex. These molecules are then either stored within the Golgi apparatus or they are released into the cell cytoplasm, where they move to the cell membrane and are released out of the cell. In addition to delivering vesicles of various cell products to different locations throughout the cell, Golgi apparatus also add proteins and sugar molecules to vesicles to serve as identifiers that ensure the vesicles are delivered to the proper area. While many of the raw products and bases used within the cell are produced within the endoplasmic reticulum, it is the Golgi apparatus that ultimately transforms those molecules into a final usable form. Given the different sacs within the Golgi apparatus, different end products can be created as the environment for chemical transformation is favorable for different reactions in different areas. Stomach acid and mucus are examples of products produced by the Golgi apparatus. While the Golgi apparatus is obviously involved in the creation of new biological polymers, it also has the ability to facilitate the breakdown of cellular waste products through the creation of lysosomes. Lysosomes are essentially the cell's housekeepers or garbage disposals that recycle the cell's organic material and digest macromolecules like nucleic acids, fats, proteins, polysaccharides, lipids, and non-functional organelles. Each lysosome contains up to 50 different digestive enzymes, first created in the endoplasmic reticulum and refined within the Golgi apparatus to become acidic hydrolase enzymes that break down complex molecules within the cells, such as sugars and proteins. Lysosomes are prevalent in white blood cells where they aid in the destruction of bacteria, dead cells, cancerous cells, and other foreign matter. As a brief summary, the cell nucleus is the brains of the cell, the endoplasmic reticulum is the manufacturing plant, the Golgi apparatus is the storage facility and post office, and the lysosomes are the cleaning crew. With that in mind, the last organelle we need to discuss is the mitochondria. To continue our analogies, the mitochondria are the batteries or powerhouses of the cell. Mitochondria are like the digestive system for the cell. They are the sites of cellular respiration, the process by which the food we eat is ultimately converted into energy for the cells. Their main function is to metabolize or break down carbohydrates and fatty acids to generate energy. The number of mitochondria within a cell will vary given the purpose of the cell. Red blood cells, for example, do not have any mitochondria as those cells need room for hemoglobin molecules to transport oxygen throughout the body. Muscle cells, fat cells, and liver cells, on the other hand, have a very high number of mitochondria given the energy needs of those types of cells. If a cell needs even more energy, additional mitochondria can be created. Mitochondria also has its own DNA and ribosomes and can make their own proteins. This DNA is a simpler form of DNA than that contained within the cell nucleus, however, in that mitochondria DNA lacks repair mechanisms that help prevent mutations. The structure of mitochondria is such that the internal surface area of the inner membrane is maximized through numerous folds called cristae, which improve the productivity of cellular respiration by the mitochondria. There is also a smooth external phospholipid membrane around this internal phospholipid membrane. Now, as we just mentioned, cells need energy to function. This energy comes predominantly from glucose, a type of sugar comprised of 6 carbon atoms. The problem, though, is that if the body broke this sugar down directly, a lot of energy would actually be lost in the form of heat, and the body cannot convert heat into energy that can be used by cellular processes. Therefore, the sugar must be broken down or converted into something that is easier and more efficient for the cells to process. The energy that would have been lost as heat by metabolizing sugar directly must somehow be captured and transported in a form that can be used by the cells. That something is adenosine triphosphate, or ATP. ATP is a high-energy compound that transports chemical energy within cells for various metabolic purposes, such as the synthesis of chemical compounds, cellular movement, and cellular division. ATP is an organic compound composed of 3 main parts: sugar, ribose, adenine, a nitrogenous base, and triphosphate, 3 phosphate groups connected by shared oxygen molecules. When water molecules break down ATP through a process known as hydrolysis, one of the phosphates is detached, releasing energy. Thus, ATP can simply be thought of as stored energy for intracellular activities. As a matter of fact, many biologists actually refer to ATP as the energy currency of life. ATP also plays a part in active transport, where materials are pumped into or out of cells across the cellular membrane. It helps maintain ideal fluid volume within the cell, and it helps send signals between cells. The nervous system is reliant upon ATP to function correctly, as are muscles and their ability to contract. So, if ATP is so important to the functioning of our cells, how is it created? That is where we need to begin talking about the process of cellular respiration. To begin, there are 2 types of cellular respiration we need to evaluate. The first occurs within the presence of oxygen, which is called aerobic respiration. The other occurs without oxygen, otherwise known as anaerobic respiration. As you will see, aerobic respiration is preferred as a lack of oxygen creates some problems, but more on that later. To summarize the process, the body takes sugar with oxygen, along with adenosine diphosphate and some phosphorus atoms, to produce ATP with additional byproducts of carbon dioxide and water. This is how that reaction looks in a chemical formula. Be sure to remember this formula, as it is very important for the proper functioning of our cells and the body. While it sounds somewhat simple, the process by which sugar and oxygen are converted into ATP is a little more complex. There are essentially 3 steps in the process of creating ATP: glycolysis, the Krebs Cycle, and the Electron Transport system. In the first step, glycolysis, one 6-carbon glucose molecule is burned, oxidized into 2 molecules of pyruvic acid, C3H4O3. This occurs in the cytoplasm without oxygen, making that process anaerobic. In order to facilitate this process, the cell must actually use 2 molecules of ATP. 4 molecules of ATP are subsequently formed in this process, along with the 2 molecules of pyruvic acid, also called pyruvate. Thus, while 4 ATP molecules are produced, 2 molecules of ATP were needed for this process, resulting in a net production of 2 molecules of ATP through the glycolysis stage of cellular respiration. This process is actually a little more complex than mentioned here, but this summary is adequate for our purposes. From there, the next step is what is known as the Krebs or citric acid of tricarboxylic acid cycle. This takes place within the mitochondria. The actual details of the Krebs cycle are somewhat complex, so what follows is an abbreviated version of the 9 steps of that cycle. To begin the Krebs cycle, the pyruvic acid formed during glycolysis is moved into the mitochondria where it is converted into a molecule called acetyl-coenzyme A, acetyl-CoA for short, releasing a molecule of carbon dioxide. In the presence of oxygen, which makes this an aerobic process, the hydrogens are stripped off of the acetyl-CoA until there are no hydrogens left. For each pyruvic acid molecule, a total of 3 molecules of carbon dioxide and 1 molecule of ATP are produced. As there are 2 pyruvic molecules created for each glucose molecule, this results in 6 molecules of carbon dioxide and 2 molecules of ATP. This production of carbon dioxide and its status as a waste product of cellular respiration is important for the paramedic, especially given discussions in later modules regarding end-tidal CO2 monitoring of patients through capnography or other mechanisms. 2 compounds capable of storing high-energy electrons, nicotinamide adenine dinucleotide NAD and flavin adenine dinucleotide FAD are also produced. These 2 compounds are reduced further into NADH and FADH2, an enzyme and coenzyme that play major roles in carrying high-energy electrons into the final step of the process. Now, this may all sound great, but if you are keeping track, these 2 steps in the cellular respiration process have actually created more waste products than ATP, 6 molecules of carbon dioxide and some water versus 4 molecules of ATP. We needed to consume ATP to create ATP in the glycolysis step, and the Krebs cycle, while it sounds impressive and is rather complex, really did not create that much ATP at all. So where does all of this energy come from? Essentially, the biggest bang for the buck, so to speak, comes from the last step in the cellular respiration process, electron transport. To understand this process, it is important to recognize that the electron transport chain is a series of protein complexes and electron carrier molecules within the inner membrane of mitochondria. The NADH and FADH2 molecules produced by the Krebs cycle transfer electrons onto this electron transport chain. As you can see on the diagram, there are 4 separate protein complexes that function to pass electrons down the chain, and there is a 5th protein complex that transports hydrogen ions back into the inner mitochondrial matrix. During this process, protons are pumped out of the mitochondrial matrix into the intermembrane space of the mitochondria. This accumulation of protons in the intermembrane space creates an electrochemical gradient that results in the flow of protons down the gradient through a process called chemiosmosis and back into the mitochondrial matrix through ATP synthase, an enzyme embedded within the membrane of the mitochondria. This movement of positively charged hydrogen ions through the ATP synthase provide the energy for the conversion of ADP to ATP through a process called oxidative phosphorylation, given the addition of a 3rd phosphoryl group to ADP, which had only 2 phosphoryl groups before the 3rd was added, thus creating ATP. The flow of electrons across the ATP synthase is not unlike water flowing over a water wheel, which generates energy to do work. In this case, the electrons produce the energy for the ATP synthase to produce ATP from ADP and a phosphoryl group. This flow of electrons also powers other vital cellular processes, such as protein synthesis. Interrupt the electron transport chain, and the production of cellular energy stops. Prolonged oxygen deprivation can be one of these interruptions, which results in anaerobic cellular respiration. We'll be discussing that in just a moment. Once these electrons are used, they are combined with oxygen and hydrogen to form water, which can then be removed from the body as another byproduct of the cellular respiration process. Now, it was previously mentioned that the electron transport step of aerobic cellular respiration is where the bulk of ATP was produced. Given our understanding of the aerobic cellular respiration process, a molecule of NADH produces 3 molecules of ATP, and a molecule of FADH2 produces 2 molecules of ATP. Going back to our original molecule of glucose, 10 NADH molecules and 2 FADH2 molecules are produced from each molecule of glucose through the Krebs cycle. Thus, if each NADH molecule produces 3 molecules of ATP, and there are 10 of those, that is 30 molecules of ATP. There are 2 molecules of FADH2 that produce 2 molecules of ATP each, resulting in another 4 molecules of ATP, bringing the total to 34 ATP molecules. Glycolysis also creates 4 molecules of ATP, but uses 2 molecules, resulting in a net creation of 2 ATP molecules, and the Krebs cycle also results in 2 ATP molecules. Adding all of these together, one sugar molecule metabolized in the presence of oxygen results in 38 molecules of ATP with byproducts of carbon dioxide and water. This ATP is critical for fueling our body cells, and the waste products of this aerobic process, carbon dioxide and water, are easily expelled by. So, after that long description of aerobic cellular respiration, what happens if cells are required to work without oxygen? How is ATP produced anaerobically? The simple answer is that glycolysis still occurs within the cell cytoplasm without any oxygen, as it does within the first step of aerobic cellular respiration, but the Krebs cycle and electron transport do not occur. Thus, 2 ATP molecules are used to produce 4 ATP molecules, resulting in a net production of 2 ATP molecules along with pyruvic acid. Without oxygen to support the Krebs cycle, however, that pyruvic acid cannot be oxidized to create carbon dioxide and NADH. Rather, the broken down pyruvate ferments into lactic acid. If a supply of oxygen is later restored to the cells, the lactic acid is easily converted back to pyruvic acid and aerobic respiration continues. If the oxygen deprivation continues, your cells will eventually reach a point where there is not enough ATP available for them to function. When this happens, the cells lose the ability to transport water and ions through the cellular membrane. The sodium-potassium pumps stop working, impacting the osmolarity of the cell, which is something we will be discussing in another module. Water will then passively enter the cell, causing it to swell and eventually burst. This kills the cell, and if enough cells die, so too does the impacted tissue, which can then lead to the death of the associated organ or organs, which can then lead to the death of the organism itself. Again, certain cells can tolerate anaerobic cellular respiration for different periods of time given varying conditions that impact how much work those cells must perform. Brain cells, for example, can tolerate approximately 3 to 6 minutes without oxygen before irreversible damage occurs. Many muscles, by comparison, can accommodate interruptions of oxygen up to several hours. As you explore physiology and pathophysiology further in other modules, the importance of cellular respiration and the critical nature of oxygen within that process will become even more evident. On a microscopic level, our cells are not always packed in tightly against each other. Rather, many cells float within water or fluids in what is known as the cellular environment. As already discussed, the cells themselves also have their own internal environments within which their organelles live. The majority of this fluid is water, and there is a lot of water within the human body. Depending upon the stage of life, the human body consists of anywhere from 45 to 75% water. Infants have more water, while the elderly have less. The amount of water within the body also varies given the particular area or organ being examined. The brain and kidneys, for example, have very high concentrations of water, often ranging from 80 to 85% of the organ's total weight. Teeth, on the other hand, are approximated to have about 8 to 10% water content. Generally speaking, there are 2 types of fluid or water compartments within the body. One is within the cells themselves. This is referred to as the intracellular fluid (ICF) compartment. There is a lot of water within our various cells, and many resources state that the water inside our cells account for almost 2/3 of the total water within the human body and somewhere around 40% of the body's total weight. In an average-sized, healthy adult, there is about 7 gallons of water within our cells. In addition to water, the intracellular fluid also has dissolved proteins and electrolytes, such as potassium, magnesium, and phosphate. We already talked about the importance of water within the cell and how too much water in a cell will actually cause the cell to burst and die. On the other side, too little water in a cell will result in the systole being too concentrated to carry on normal cellular activities, which is also a problem. Outside our cells is something known as extracellular fluid, ECF. This fluid has two primary components: plasma, which is the fluid component of our blood, and interstitial fluid, which surrounds all the cells that are not within the blood. We will find out more about the body's plasma when we examine the circulatory system in greater detail. For the time being, think of plasma as a fluid within the body's circulatory system. It accounts for approximately 20% of our body's extracellular fluid and is responsible for transporting blood cells, proteins, electrolytes, nutrients, gases, and waste materials. In terms of this content, plasma contains a lot of sodium, chloride, bicarbonate, and protein. Our interstitial fluid also has high concentrations of sodium, chloride, and bicarbonate, but not as much protein. It is through this interstitial fluid that gases, nutrients, and waste materials travel between the capillaries and the cells. The extracellular fluid compartment also contains lymph, which is a clear to yellowish watery fluid that works with the body's lymphatic system to pick up fats, bacteria, and other waste products for disposal or elimination from the body. We will explore this extracellular fluid in greater detail within the module on the lymphatic and immune system. There are also other water-based extracellular fluids within the body. Synovial fluid keeps joints lubricated. Cerebrospinal fluid surrounds and provides cushioning and nutrition for the brain and the spinal cord. Peritoneal fluid lubricates the abdominal wall and the organs within the abdominal cavity. Pericardial fluid surrounds the heart within the pericardial sac. Pleural fluid serves as a lubricant between the parietal and visceral layers of the pleura surrounding the lungs, and the aqueous humor is the transparent fluid within the eye. In comparing both intracellular and extracellular fluids, there is one thing that physically separates them: the cellular wall, or membrane. As we have already discussed briefly and will discuss more in just a little bit, there are times when it is critical for various substances to move across the cellular membrane either into the cell itself or out of the cell into the extracellular fluid. Sometimes these substances are electrolytes, lipids, or proteins. Other times it may be water itself that is moving across this barrier into or out of the cell's intracellular fluid. This leads us to a discussion on solutions. Water is often a solvent in which something else is dissolved. This something else is called a solute. Take salt, for example. When you mix salt into water, the water is the solvent and the salt is the solute, which creates a solution we call salt water. As paramedics, it is important to consider the health of individual cells, which can often be impacted by the amount of intercellular fluid, predominantly water, that is within that cell. If the cell has too much water, it can burst and die. If there is not enough water within the cell, it will shrivel or shrink. Therefore, when the internal cellular environment, the cell's intracellular fluid, is compared to the external cellular environment, the extracellular fluid, the concentration of solutes in the intracellular fluid compared to that within the extracellular fluid can create a disparity that classifies a solution either within or outside the cell as isotonic, hypotonic, or hypertonic. In this particular diagram, if a container has an equal portion of solute to solvent, the solution is called isotonic. If the solute exists in a higher concentration to the solvent, that solution is hypertonic. If the solute exists in a lower concentration to the solvent, the solution is hypotonic. When we look at this within a cellular context, we begin comparing the intracellular fluid to the extracellular fluid. Let's use sodium as our solute. If a cell has a greater concentration of sodium within it compared to the extracellular fluid around that cell, the intracellular fluid is hypertonic compared to the extracellular fluid, which is hypotonic. If the concentration of sodium is the same between the intracellular fluid and the extracellular fluid, the two fluids are isotonic in comparison to each other. This terminology will come into play in just a little bit when we discuss cellular transport mechanisms. Another very important thing to understand with regard to our bodies in the cellular environment is what is known as the body's acid-base balance. If you try to remember back to a high school chemistry class, acids are substances that release positively charged hydrogen ions, protons, in an aqueous medium, water. Acids often have a burning odor, a sour taste, and are somewhat sticky. Bases, also referred to as alkalis, on the other hand, release negative hydroxide ions that bond with hydrogen ions. Stated another way, bases are proton acceptors. Bases taste bitter, are usually odorless except for ammonia, and are slippery, or, as some describe them, soapy. To measure the amount of hydrogen ions within an aqueous solution, a pH value is used. The scale goes from 0, which is highly acidic, to 14, highly basic or alkaline. If there are an equal number of hydrogen and hydroxide ions in a solution, it is said to be neutral with a pH of 7. For comparison, concentrated hydrochloric acid has a pH of 0. Stomach acid has a pH of 1 or 2, coffee has a pH of around 5, and even milk is slightly acidic at a pH just below 7. Baking soda has a pH somewhere between 8 and 9, hand soap is around 10, bleach and oven cleaner is between 12 and 13, and concentrated sodium hydroxide is extremely basic at 14. Remember that pH measures concentration of hydrogen ions in a solution, and with a change of 1 on the pH scale, there is a tenfold change in the number of hydrogen ions in the solution. So why does this matter to the paramedic? The reason is that the concentration of hydrogen and hydroxide ions within the body can affect chemical reactions that control certain physiologic functions, and the body itself maintains a specific pH that is slightly alkaline at 7.35 to 7.45. A deviation from this normal can be very detrimental to the body and its cells. While a pH of 7 is considered neutral, the body is used to being slightly alkaline. Therefore, if the pH within the body goes below 7.35, the body will suffer from something known as acidosis. If that pH falls below 7, a coma may result. Prior to that, fatigue, confusion, headache, lack of appetite, and other symptoms may present themselves. Inversely, a blood pH that exceeds 7.45 is defined as alkalosis, and a pH above 7.8 will typically cause prolonged, uncontrollable skeletal muscle contractions. Other symptoms that may occur prior to that point include dizziness, confusion, hand tremors, numbness, or difficulty breathing. As the pH level in the body is so important, it should come as no surprise that the body has mechanisms by which it maintains an ideal pH of 7.35 to 7.45. These mechanisms are referred to as buffer systems, and they work by chemically neutralizing excess acids or bases within the body. With this in mind, it should be no surprise that the body is very sensitive to changes in its internal pH. For this reason, the body has several buffer systems that work in concert to maintain an ideal pH environment within the body. There are short-acting chemical buffers in the blood that can impact the body's overall pH within seconds. The respiratory system impacts pH over the course of a few minutes by exhaling carbon dioxide and acid. Longer-acting changes over the course of hours to days are facilitated by the renal system, which controls acid-base balance by excreting hydrogen ions while conserving bicarbonate. As far as blood-based buffering systems are concerned, the most prevalent is the protein buffer system. Proteins, which are made up of amino acids and contain both positively charged amino groups and negatively charged carboxyl groups, can bind hydrogen and hydroxyl ions, thus functioning as a buffer within the body. In an acidic environment, the carboxyl group takes up excess hydrogen ions. When alkaline, a protein is released. This protein buffer system actually accounts for 2/3 of the buffering power of the blood and most of the buffering within individual cells. Another system uses phosphates that are in the blood. Sodium dihydrogen phosphate is a weak acid, and sodium monohydrogen phosphate is a weak base. In an acidic environment, the sodium monohydrogen phosphate accepts the hydrogen ions, thus taking a strong acid and a weak base to produce a weak acid and salt. Inversely, in an alkaline environment, the sodium dihydrogen phosphate accepts the excess hydroxide ions. This combines a strong base with a weak acid to generate a weak base in water, which can be excreted from the body through the kidneys. Hemoglobin, which carries oxygen to our cells, also plays a part in the buffering system by releasing oxygen and binding to carbon dioxide and excess hydrogen ions. The last set of buffering pairs is a carbonic acid bicarbonate buffer system. If you remember from earlier in the module, aerobic metabolism takes sugar and oxygen to produce ATP, carbon dioxide, and water. The body uses this carbon dioxide and water, along with the enzyme carbonic anhydrase, to produce carbonic acid, H2CO3. Bicarbonate, HCO3, is a derivative of carbonic acid that is created by a proton transfer reaction in ratios of 1 carbonic acid molecule to 20 bicarbonate molecules. The reason for so many bicarbonate molecules is because the products of cellular metabolism are acids such as lactic acids and ketones. When the body becomes acidic, this system will take carbon dioxide from the carbonic acid to be exhaled while more bicarbonate binds with protons. Inversely, in an alkaline environment, the system forces more carbonic acid to dissociate while also excreting bicarbonate ions through the urine to decrease the pH levels in the body. As already alluded to, the respiratory system also plays a part in regulating the acid-base balance in the body by expelling or retaining carbon dioxide, which is an acid. To increase the body's pH, the respiratory system increases the respiratory rate, which causes the person to exhale more carbon dioxide over time, making the body more alkaline. Inversely, slowing the respiratory rate or trying to re-inhale exhaled carbon dioxide by having a person breathe into a bag will decrease the individual's pH, making him or her more acidic. Lastly, the renal system addresses the metabolic component of the buffering system by controlling the levels of bicarbonate in the blood. Only the renal system can regulate alkaline substances in the blood and restore chemical buffers that are used in managing hydrogen ion levels in extracellular fluids. While the respiratory system can eliminate carbon dioxide, it cannot expel other acids generated by cellular metabolism, such as phosphoric, uric, lactic, and ketone acids. That is another job of the renal system. If the body becomes acidic, the renal system retains bicarbonate and excretes hydrogen ions into the urine through hydrogen phosphate and ammonium. If the body is alkaline, the renal system retains hydrogen ions and excretes bicarbonate. Again, this is a slow buffering mechanism that does its work over the course of hours and days. All of these various systems work together to help the body maintain a healthy acid-base balance in the 7.35 to 7.45 range. In later modules, you will learn more about problems associated with being too acidic or alkaline and what the paramedic can do about it. As we talk about moving things into and out of living cells, whether that be water, proteins, electrolytes, or whatever, remember that each cell is contained within and protected by its own cellular membrane, and these various molecules cannot move back and forth across that membrane at will, so to speak. There must be some type of driving force or mechanism that allows these various molecules to move into and out of the cell. Thus, our look at cellular transport mechanisms. The first cellular transport mechanism we will examine is diffusion. Diffusion is a process of particles or molecules moving from an area of high concentration into an area of lower concentration to try and even out the concentration between two areas. Essentially, the molecules within a container want to fill the available space, and if there is a membrane that separates two fluids of different solute concentrations, those solutes will move across the membrane, if they can, to equalize the concentrations on both sides of the membrane. This is a passive process that does not require energy to accomplish. This diagram shows the process of diffusion, where there is initially a high concentration of a certain molecule outside the cell, and over time, some of those molecules enter the cell until the concentration is equal both inside and outside the cell. This process also works the other way, with molecules leaving the cell if the concentration is higher inside the cell than outside the cell. If it helps, think of what happens when you drop some food coloring into a glass of plain water. Over time, the food coloring diffused through the water so that all the water is equally colored. Another example would be how the carbon dioxide in a carbonated soft drink will escape into the atmosphere over time, leaving that beverage flat. Have you ever seen a helium-filled balloon deflate over time? That is another example of diffusion, as the helium slowly diffuses through the balloon itself into the surrounding atmosphere. Within the body, this process of diffusion is prominent in the lungs, with oxygen and carbon dioxide moving across the alveolar wall and the capillaries. It also occurs in other organs as well, such as the kidneys, the small intestines, and even the cornea of the eye, to name a few. Another passive transport mechanism is osmosis. Rather than the solute moving as it did in diffusion, however, the solute itself, such as water, moves from an area of low solute concentration to one of high concentration. An example of how osmosis works is provided in this illustration. If water were in this U-shaped tube all alone without the membrane on the bottom, one would expect the water level on both sides of the U to be of equal heights. In this illustration, however, there is a semi-permeable membrane at the bottom of the U that separates one side with plain water from the other side that contains some type of concentrated solution. Because the solute cannot move across the semi-permeable membrane to reach equilibrium, the water itself, which can pass through the barrier, moves to the area of higher solute concentration to try and even things out, so to speak. To help further, let's say that the concentrated solution on the right side of the U in the illustration is salt water, and the left side of the U contains plain, non-salt water. Because the salt cannot move across the semi-permeable membrane, but the water can, the water will move through the membrane to try and equalize the salt concentration on both sides of the membrane. This is where the hypertonic and hypotonic definitions come into play that we discussed earlier. The right side of the U is hypertonic, given that the concentration of the solute, the salt, is higher on the right side than the left. Inversely, the left side of the U is hypotonic, as there is a lower solute concentration, less salt, on the left side than there is on the right. Another passive transport mechanism exists to move molecules from areas of higher concentration to lower concentration. Remember our discussion on how the cell membrane is made up of a phospholipid layer that allows fat-soluble substances to move across the membrane while other molecules are prevented from making the same journey? To allow for the movement of these molecules that cannot easily pass through the cell membrane on their own, such as glucose, sodium ions, chloride ions, and so on, a process of facilitated diffusion is used. One way facilitated diffusion works is by binding the otherwise excluded molecule with a carrier protein located within the cell wall. The protein changes shape to allow the transport of the substance, and then the protein returns to its original position and the molecule is now where it wants to be. This is how glucose, which is a rather large non-lipid soluble molecule, moves across cell membranes. Another way facilitated diffusion works is through protein channels in the cell membrane that allow smaller ions to pass through the channel across the cellular membrane, similar to a tunnel through an otherwise impassable hill or mountain. These channel proteins are water-filled pores that allow polar molecules, water-soluble molecules, to move through the non-polar, lipid-soluble interior of the cell membrane bilayer. To control the flow of ion exchange through the cellular membrane, many of these channel proteins are gated, and they remain closed until they receive a special chemical or electrical signal that opens them. There is also a class of channel protein called an aquaporin that allows the rapid movement of water molecules through the cellular membrane. Thus far, all of our mechanisms for moving molecules across a concentration gradient have been passive in nature. Essentially, molecules have a natural tendency to move from an area of high concentration to low concentration through diffusion. That, or water itself, will move from an area of low solute concentration to an area of high solute concentration to try and equalize the concentration of the solute between the two different areas through osmosis. Again, given the passive nature of either diffusion or osmosis, no energy is used to make this movement of molecules or water happen. Sometimes, however, it is necessary to move molecules from an area of low concentration to an area of high concentration. This does not occur naturally through diffusion or osmosis because doing so creates an even greater concentration gradient. Therefore, to move these molecules across the grain, so to speak, we need to actively transport them across the cellular membrane from an area of low concentration to an area of high concentration. To do this, we once again use carrier proteins, but we must add energy, ATP, to actively move or transport the molecules. Given the active nature of this process, the proteins are often referred to as pumps, or protein pumps. One type of protein pump is known as an antiport pump. which moves one substance one direction while moving another separate substance in the opposite direction. These pumps are very efficient as they can typically use one molecule of ATP to facilitate the movement of two separate molecules in different directions simultaneously. A good example of an antiporter is the sodium-potassium pump, which is used by the body to transport potassium into cells while moving sodium out of cells to facilitate the firing of nerves and contraction of muscles. Without this active transport mechanism, we would not survive, given how important the proper functioning of nerves and muscles is to the body. It has been surmised by some that 20 to 25% of the energy we obtain through our food is consumed within the body to keep these sodium-potassium pumps working. Another type of protein pump is a symport pump. This type of pump uses a molecule that wants to move from an area of high concentration to an area of low concentration as a carrier of sorts for another molecule that needs to move against its concentration gradient. Sodium-glucose transport proteins are examples of symporters that use two sodium ions which want to enter the cell as a carrier for one glucose molecule which would want to stay out of the cell. While ATP is not expended to move sodium into the cell along with the glucose it is carrying, ATP is still expended elsewhere by sodium-potassium pumps to maintain the sodium gradient between the intracellular and extracellular environments that actually allows the sodium-glucose pump to function. Additional forms of active transport are endocytosis and exocytosis. When substances are too large to enter or exit the cell through the cellular membrane, the cellular wall itself will be modified to facilitate the movement of these substances into or out of the cell. In endocytosis, the cell wall changes to trap substances or molecules, create a vacuole or vesicle, and pull them into the cell. There are actually three types of endocytosis. One is pinocytosis, which is used to move extracellular fluid into the cell. The second is phagocytosis, in which a cell surrounds a foreign particle and engulfs it, which is very common practice in the immune system. The last is receptor-mediated endocytosis, in which cellular membrane proteins are utilized to create a pocket in the cell membrane that has receptors that capture specific substances. This pocket is eventually closed off, creating a vesicle within the cell that contains the substances that were previously outside the cell. Exocytosis is a similar process in reverse. When a cell wants to move something outside the cell, such as an enzyme, hormone, or antibody, the cell will create a vesicle to enclose that substance. That vesicle will then move to the cell membrane and fuse with the membrane, eventually becoming part of the cell membrane. When that happens, the vesicle opens and the contents are released. This is the process used when protein products are made in the cell's endoplasmic reticulum and packaged in vesicles. These vesicles are sent to the Golgi apparatus, which adds molecules that will be recognized by receptors on the membrane of the cell before packaging these products in a vesicle of its own that will move to and fuse with the cellular membrane for release of the contents into the extracellular space. Lastly, while not necessarily a cellular transport mechanism in and of itself, filtration does play a part in the selective movement or lack thereof of substances across membranes given the application of hydrostatic pressure from the cardiovascular system. The kidneys provide a great example of filtration at work as blood is essentially filtered through the kidneys to remove water, salts, glucose, and urea for excretion by the body through urination. Don't worry, the functioning of the kidneys will be discussed in much greater detail in later modules related to the renal system. As the building block for living organisms, it should come as no surprise that cells themselves have a specific life cycle. Cells are created from other cells. They perform their routine functions and eventually die. Additionally, for an organism to grow, the cells of that organism must either expand or multiply. For the time being, we will not worry about cells increasing in size. Rather, we will focus specifically on cells replicating themselves or multiplying. There are two processes by which a cell can divide, thus creating more cells to facilitate growth or replace dying cells: mitosis and meiosis. Both processes start with a single parent cell and have similar steps for producing new cells. The biggest difference between the two is the propensity for genetic variation given the application of sexual reproduction. In mitosis, the parent cell divides to produce two identical daughter cells. This is done in 5 recognized stages: interphase, prophase, metaphase, anaphase, and telophase. When mitosis is occurring, all of the cell's energy is used for the process of cellular division. In brief, chromosomes are replicated and compacted within the nucleus of the cell, and the nucleus membrane dissolves, releasing the chromosomes into the cell. The chromosomes line themselves up end to end across the center of the cell, and spindle fibers, which extend between the centrioles that are now on opposite sides of the cell, begin to attach to the sister chromatids that are within the chromosomes. During anaphase, the sister chromatids are pulled apart by the mitotic spindle to opposite ends of the cell. These full sets of chromosomes gather together at each separate end of the cell, and a new membrane forms around each of these 2 sets of chromosomes while they lengthen and unwind to create 2 new nuclei. In telophase, the original cell then goes through a process called cytokinesis, where the parent cell is essentially pinched in the middle to separate the cytoplasm of the parent cell into 2 new cells. Where one cell existed previously, there are now 2 new cells that are identical to each other. Meiosis, while it follows a somewhat similar process, produces 4 cells from a single cell that divides twice. Each of the 4 daughter cells have only half the number of chromosomes of the parent cell. This occurs over a 9-step process where the interphase is similar to that in mitosis, but then there is a first prophase, metaphase, anaphase, and telophase, resulting in 2 cells, followed by a second prophase, metaphase, anaphase, and telophase, as the 2 cells are then divided again into 4. Again, unlike mitosis, only 1/2 of the parent cell's genetic code is copied into each of the resulting 4 cells. Each daughter cell has half the number of chromosomes as the parent cell. Given your knowledge of the human body from the modules you have completed thus far, you are aware that the body is filled with cells that perform specific functions, such as muscle cells, nerve cells, blood cells, and so on. These cells are referred to as specialized or differentiated cells. With rare exceptions, such as liver or T cells, differentiated cells cannot divide to make copies of themselves. For the most part, only cells known as stem cells can divide and copy themselves. When stem cells split, they either create more stem cells or more specialized, differentiated cells. Therefore, it is important that stem cells not only produce differentiated cells to replace damaged or dying cells, but they must also self-renew, which means creating identical copies of themselves as well. If stem cells did not copy themselves, the body would run out and, because differentiated cells typically cannot undergo mitosis, the organism would die, as damaged or dying cells would not be replaced. As far as stem cells are concerned, there are embryonic stem cells that exist shortly after conception in early embryo development, before the embryo has even implanted into the uterus, and then there are tissue stem cells that exist in a developing fetus, babies, children, and adults. Embryonic stem cells can develop into virtually any tissue that will be a part of the newborn baby, while tissue stem cells can only differentiate into the kinds of cells found in the tissue to which they belong. Thus, muscle stem cells can only produce muscle cells, blood stem cells can only make blood cells, and so on. In 2006, scientists discovered a way to develop stem cells in a laboratory setting that behave like embryonic stem cells. These laboratory-created stem cells are called induced pluripotent stem cells, and some believe these iPS cells may be the key to treating some diseases like Parkinson's or Alzheimer's. There are obviously a lot of cells within the human body, and it is critical that those cells, when they divide, produce identical copies of themselves, which includes the critical DNA contained within them. If that DNA sequence is not copied perfectly, either due to a copying mistake within the cell or due to external factors such as cigarette smoke, UV light, or radiation, the resulting DNA sequence is referred to as a mutation. It is no longer a perfect copy of the original DNA sequence. In some cases, the mutation may actually be a good thing that leads to genetic variation within the population or is responsible for some type of trait or attribute that proves to be beneficial to the organism. Sometimes these mutations can be both good and bad, such as sickle cell anemia, which is caused by a mutation in the gene that instructs the building of hemoglobin, resulting in red blood cells with an abnormal, rigid sickle shape. Sickle cell anemia can result in pain, fatigue, swelling of the hands and feet, infections, delayed growth, and vision problems. On the other hand, it also protects against malaria, which is prevalent in some areas of Africa, which is why sickle cell anemia is typically associated with people of African descent. Mutations can also be very bad by disrupting normal gene activity and causing diseases such as cancer, which you will learn about in a later module. One of the things that differentiates a paramedic from an EMT is the paramedic's knowledge of how the human body works at the cellular level and how paramedic interventions impact the cells and their functioning. While the cellular environment and functioning is even more complex than discussed in this module, you should now be able to discuss cell theory, describe cellular anatomy and physiology, explain cellular respiration, describe the cellular environment, describe cellular transport mechanisms, and discuss the process of cellular division at a level appropriate for a paramedic. As you proceed further into your paramedic training and education, the information within this module will serve as a foundation for additional learning. This presentation was prepared by Waukesha County Technical College in Pewaukee, Wisconsin, and is distributed with an Attribution Non-Commercial Share-alike 4.0 International Creative Commons license. Copyright 2020 Waukesha County Technical College. For information on WCTC's numerous fire and EMS educational offerings, please visit us online at wctc.edu.